I am really honored. Um, this particular topic is near and dear to my heart, having had a family situation with it. And when I read about the work of Dr. Amoncolor, um, I just felt like we really needed to hear from him. You all probably know that he's the chief of neurosurgical oncology and the director of the Penn Brain Tumor Center, and he's won awards from NCI. And I am going to just turn it over to him because this is a huge topic.
Well, thank you so much. It's a first of all, it's a pleasure to be here, I feel like having arrived into 2021 at the heart of COVID, it's almost like I'm starting anew. You're starting to like get to meet people, and now that we're opening up a little bit, so I'm glad that I can speak to members of the community because there's just so many. The great thing about Penn and its translational capacity is that I get to work with like Drew Weissman, who's like, I want to work on GBM, and I'm like, I almost passed out when he said that. It's like you're going to work on GBM, and that community is really incredibly important. That's why I came here. And I have to say that this is the right time to sort of start building this nucleus. As Lucy introduced, I'm Nduka Moncolor. I am a neurosurgeon scientist. She wanted me to say a couple of words about myself and my sort of career, but I really, was very interested in neuroscience from a very early age. And you'll see some biographical details, along the way that kind of caused me to think about brain tumor surgery a little bit more carefully. But I kind of always wanted to be a neurosurgeon. And then I found that really there's so many unsolved problems, with regards to the brain that it was a natural fit to focus on science, and I'd always been interested in science. And so when I got to med school at Yale, I worked on vascular genomics, which would prove really sort of poignant and a starting point for my interest in genomics. And then I decided to do my neurosurgical residency at Yale. But during that time, I spent 2 years in the lab of one of the luminaries in neuro-oncology research by the name of Eric Holland. And Eric put me on a project where he decided that I was going to work on brain injury, and I was like really upset, because I'm here to do brain tumors. You want having me work on brain injury, but it also proved to be serendipitous because through that experience, I started to learn immunology, because brain injury and the recovery from brain injury has a lot of immune components, and the brain being thought of as an immune privileged organ. You can clearly see that was not always true. And so my career then really is I think this talk captures all elements of my career, which includes obviously the surgical care of patients with high grade gliomas and brain tumors, but also translating over to science. I am at my core, I would say, an immunogeneticist, and that is something that I think has served me well, because understanding both the genomic basis of disease and the immunological consequences, I think, is I believe the only thing that will solve brain tumors. Let me see if I can. So the part one of this talk, I entitled it "Genomics of Cancer Cells and the Brain," and it's just a very introductory sort of evaluation of how genetics influence cancer development in the brain, particularly for primary brain tumors like glioblastoma, and also a subset of tumors called diffuse gliomas, which inevitably go on to be glioblastoma-like. And that's actually what I've kind of focused my career on. But at the core of cancer is genomics, right? So if, to ignore genomics is to ignore cancer. And so I think that's what I'll start with. But I have to say, I don't have any disclaimers, but my one primary disclaimer is that I have some skin in the game. My dad, who died of glioblastoma in June 1994 when I was, I think I was like 15 or something, 15 or 16. And was like a real role model. This guy, grew up in a small village in the middle of Nigeria, and his dad was a small game hunter who used a catapult and stones to kill small animals and then sell them. And somehow his son became a professor and a true intellectual who, till today, is probably the smartest person I know. And I'm not just saying that. I mean, the guy was a real genius, and there's actually a weird correlation between intelligence and glioblastoma, which we still haven't figured out. But he went on to be a professor at UCLA and then ultimately at NYU, where within a couple of months of getting to NYU and moving to New York reluctantly, because I lived in Santa Monica, it was beautiful. And my dad came along and was like, "You got to move to New York." So we moved to New York, and within 3 months, he was diagnosed. He had some headaches. He was diagnosed with a 3 centimeter left temporal GBM that was resected. And this is a point, I think, even though I don't have the films, I know almost certainly that it was resected subtotally and in a manner that did not give him the best chance to do to maximize his overall survival, however short that would be. And it turned out to be incredibly short because he was dead in 7 months on January 9th, 1995. So this had an impact on me, but obviously I sort of went about my life, and I thought that I was still interested in neuroscience. I really didn't want to do neurosurgery, but the more I sort of went through med school, it came down to neurosurgery and neurology. And to be completely honest, I didn't like to round. I mean, rounding was just one of those things where I thought was like a little excessive, and so neurosurgery then became the path. And the first time I went to neurosurgical OR. I saw an aneurysm being clipped, and I'm not a super, I might be spiritual, but I'm not exactly religious, and that is as close as I've come to having a true sort of religious experience and seeing this aneurysm clipped and like the beauty of the brain. And so I was totally sold from there, and I thought for sure I was going to do vascular neurosurgery and clip aneurysms for the rest of my life, but there's always, I guess, your subconscious remembers, and as I treated patients with GBM in residency, I just realized that there was a lot more to do. So it's the most common primary brain tumor in adults. It is what Eric Holland used to call the Terminator. It's invasive and highly vascular. The average overall survival, even even today, is 12 to 18 months. Now we've managed to push that at an institutional level at certain places by recognizing some elements of that I'll talk about a bit later. But the 5-year overall survival is still less than 5%. Then this is academic data. This is not even looking at patients who are treated at private hospitals, where the overall outcome is substantially worse and has been documented. So this is what I call the infamous family of gliomas. As you'll see, starting from left to right, you get more aggressive. Kids get a type of tumor called a pilocytic astrocytoma. which is entirely curable with surgical resection. There's another sort of mildly diffuse tumor called ganglioglioma, and that is also curable with tumor resection just because it doesn't grow beyond a certain point. So the goal there is kind of controlling seizures, which patients get. And then after that, you really get into the true infamous family of gliomas, right? So WHO and WHO grade 2, grade 3, and grade 4 tumors. They start with oligodendrogliomas, which is sort of the luckiest you will get with a diffuse glioma. The overall survival is probably about 15 years or so with great care. And then astrocytomas where the overall survival, WHO grade 2 astrocytoma where the overall survival is about 7 months. And then they, as a tumor transforms, you get anaplastic versions of those two, and then you get a GBM version of that of this diffuse glioma. And this is really what I've spent the early years of my career studying. And then you get into WHO grade 4 tumors, which are usually have a signature that does not feature a mutation in a gene called IDH. These tumors, the overall survival, as I said earlier, is about 14 months. And then, for the really unfortunate, there's a very diffuse glioma that we don't understand very well that actually myself and one of my colleagues are beginning to work on, because it's just like they often kind of look like low-grade gliomas in their sort of radiographic appearance, but they are just hyper-invasive. And so what makes them, hyper-invasive, we don't really understand. That's called gliomatosis cerebri, and 9 months is really an overstatement. I mean, these patients can sometimes die within, days or months of discovery of the tumor. I always obviously like we've got a couple of Nobel laureates at Penn, but I always say that the transformative event in glioma biology, and the thing that really made me think that we were on our way to doing something, meaningful, was that the NCI designated the Cancer Genome Atlas, and obviously GBM was the first tumor studied, and this led to, for all of us who, I was at the end of med school when the TCGA data first came out, or actually, no, I was in residency, early residency when TCGA data came out, and for sure you thought, okay, we've figured this out. We are going to cure this because we know the genes, all the genes that are altered in GBM, and so it's all about, targeting these genes, and for some of them there are targets, but unfortunately that's not changed much. But what the genomics tell you about at least GBM is that in general you require 3 events, right? So first you require growth factor alteration, so something that allows tumors to grow without the normal level of nutrients and growth factors that are required for cells to proliferate. And so EGFR is by far the most common of those events, although EGFR is as a mutation, as a driver, really has a diverse phenotypes depending on the co-mutations that occur and whether or not the tumor is methylated or not. And then you need oncogenic pathway alteration. So this involves for the most part the most common event in that pathway is TP53 mutations, which are the most common mutations across the entire glioma landscape. And this is they're altered in about 87% of all gliomas in one form or another. Either the inhibitors of p53 are lost, or TP53 itself. The inhibitors are the TP53 itself is lost, or the the inhibitors of TP53 are also mutated in some form of MDM2 and MDM4. So together, that really captures 87% of all gliomas. And then you have cell cycle activation, which is a common feature of higher grade gliomas. So these three events really need to coordinate to form IDH tumors. And to simplify all this, I started thinking about it like, building a house, right? So like if you build a house, you need the initial events, that form your baseline. So that's your foundation. And at the foundational level of diffuse gliomas, which as I mentioned earlier, and I'll talk about more, are lower grade tumors at discovery, but they inevitably always go on to be high grade tumors. So they've IDH1, IDH2 form the foundation of diffuse gliomas. And at the foundation of true glioblastoma, that is primary glioblastoma, are these TERT promoter mutations. And so that's a foundational level, right? And then as you build a house, you gotta get to the first floor. And p53 is one of those mutations that is ubiquitous and is really found in both glioblastoma and in diffuse gliomas. And that's what's required to form the first floor where the tumors become a little bit more proliferative and aggressive. But if I have a tumor that I'm resecting that turns out to be only have IDH and p53, and I've taken out, I've achieved a gross total resection of the visible tumor, that's a huge win. Those patients actually go on to live sometimes for like 30 years. I've seen a few 30, 35-year survivors of those types of tumors. But for glioblastoma, once you get to first floor, that's where the trouble starts. And when you get to second floor for both diffuse gliomas and glioblastoma, where you have common mutations usually associated with cell cycle alterations, these tumors really take off. And so, I think there's a very sort of, I think, tangible and way of describing how to build a glioma. And of course, there are multiple other variables that I don't talk about, but because of this interest in genomics, and because of my interest in brain tumor surgery, I really started to think, how do these tumors, how do the genetic influences affect what I do in the operating room? So, and this was for the first part of my career, I was really, all about, translational science, and I kind of poo-pooed any kind of clinical research. I thought that was for the people who couldn't do any basic or translational science. But then you find that there's some meaningful questions, and often those meaningful questions are directed back at yourself as a surgeon, which is ultimately for us, it was like, how do these genomic alterations affect what we do in the OR? To what extent do we have to be aggressive or non-aggressive? And I love this quote from Charles Leclerc, or Leclerc, as someone who loves Formula One. He says, being honest with myself is something I like. I'm happy I don't make excuses when I make a mistake. This is the fastest path to progress. And this is really I think the motto that I live by. And the reason for that is that, as if we didn't already know, brain tumor surgery is incredibly hard. It's like not just that. I think it is something that everything you have a very, very fine line between wrecking a human being and making them well. And so whenever you kind of think about how much effort that's involved in resecting a brain tumor, I always compare it to like breast cancer, where the ability, where the effort in breast cancer is obviously not something that we ever think of as not significant, but what you get out of a complete lobectomy a lumpectomy, is quite significant. You get pauses, just making some weird noises. But what you get out of something like breast cancer, or increasingly a melanoma biopsy, or or even a complete resection of a melanoma, where there's still some remnants that you can't get. There's far more efficacy, right? For in melanoma, breast cancer, increasingly lung cancer. I mean, so we're still sitting here as brain tumor surgeons, proud that we're neurosurgeons, but at the end of the day, you have to, again, be honest with yourself and think to yourself, well, what am I really accomplishing? And it's a big deal to give someone, a few years of life. I think every day of life is meaningful, but I think if you really think about it, this is fodder for real in-depth analysis of what you do, and looking at it from multiple different angles. And so that I had 3 students when I was at Pitt that where we began with the question, well, number one, GBMs are heterogeneous, right? And we have very good literature in the field saying that the maximum extent of resection of the contrast enhancing disease, that is, if you have a T1 MRI sequence, all the tumor that you can basically see that's contrast enhancing, if you maximally resect that, you get the best outcomes. But that's a population level sort of description. But are there any nuances, right? And what we wanted to ask was, how do the genomics influence that? Does the genomics alter the amount of residual tumor that you can leave? Are there tumors where you have to take out all of it? Are there tumors where you have to do a super total resection? Are there tumors where taking out too much is potentially harmful? And so that was the idea. But behind this idea, I think it's like a really a sort of a deeper sort of network of events, right? So you've got the tumor and its genomic drivers, which then drive these genotype specific immunologic states, right? And on the surgical side, you resect this tumor that's volumetrically defined. You have your postoperative outcomes, because if you hurt a patient, they don't do as well. And then, but all these factors interact, right? So if you have a tumor that happens to have some immunogenic qualities, right? Then does it make sense to resect that entire tumor, or do you want to leave a nodule of the tumor that can react, like that forms a basis for an immune response, right? And this is really something that we don't know, right? And so this is the question we wanted to answer, because at face value is just to talk about genomics and resection, but at a deeper level, there are so many basic translational scientific questions that are at the heart of that. So what we did is we used a Monte Carlo based analysis to look at a very, very large cohort of GBMs that had next generation sequencing, and then we validated that cohort with GBMs from Penn when I got here, and I'm happy to say we then extended that to a cohort that's not here at UCSF, where we got another 500 tumors from UCSF. And there's a paper that's almost completed, and we're sending out to JCO, so hopefully they understand the impact. But essentially what we did is we use this minimally selected log rank statistic to find the right, or the volumetric cut point at which you had the best chance of survival, which that population had the best chance of survival. It was a little different way of looking at it, because normally we say, okay, well, you look for whether or not a patient who's had 100% resection versus a patient who's had 93% resection, if one is better than the other. But really, when you look at these individual genetically defined populations, the question here is, do they have different volumetric cut points? Meaning, to what extent do you have to resect all of them to get a meaningful surgical benefit? And so by this sort of largely unbiased clustering, we came up with four real or five real tumor cohorts or genomic cohorts that we could analyze. And one was interestingly EGFR mutant tumors, or any EGFR pathway alteration was fit into one category. There was a p53 mutant population that really lacked a lot of other genomic alterations and formed its own unique cluster. There was an other, these are within the methylated subclass. So I'll take a step back and say that in general, one of the hallmarks of GBM is that tumors that exhibit methylation in a gene called MGMT generally are thought to do better, whereas unmethylated tumors do worse, right? And we've, there's been a wealth of sort of sort of basic scientific explanations for that, none of which are satisfactory to me. And this data kind of shows you why that the difference between methylated and unmethylated is sometimes not it can be sometimes arbitrary, because if you look at EGFR mutant tumors, whether or not they're methylated is really important, but there's another cohort of tumors that happens to be methylated that is not quite as favorable. And so in general, the unmethylated group had the most patients. Notably, and I think what was meaningful is that we did not have a significant difference in tumor volume across the entire cohort. So there's no statistically significant differences. There was, there was some mild differences in age at diagnosis, but and also in sex, but that was those were the only real differences. But when you look at the multivariate analyses, these differences largely disappeared. And so to go in further to this is really kind of the heart of the data. And what it shows is that the cut points at which you get the highest amount of survival is remarkably different for all of these genomic subtypes. And this is not something that we were expecting. So, overall, our overall survival was, certainly better than average. And this is a great data set because it captured tons of different surgeons, some of whom were not true neurosurgical oncologists, and so we were able to get a wide variety of tumor volume that was left behind. Whereas if you go to a true surgical oncologist who's been doing this for a really long time, they've been trained to remove every ounce of tumor. So the question then became, what do these cut points mean? So I'll take your attention to D and start at D. So as I told you before, MGMT methylated tumors generally are thought to be favorable, but you found that this MGMT methylated other group, which actually the other turned out to be mostly CDKN2A and B, which is one of the cell cycle pathways that I talked about earlier. And with this tumor, you could basically leave nothing. I mean, 0.1 cc's. I mean, that is not really perceptible, at least surgically. But it may, but the difference between patients who did not have up to 0.1 cc's and who had more than 0.1 cc's of residual, and those who had less was remarkable in that if you had less than 0.1 cc's, or essentially a gross total resection, your median overall survival was greater than 2 years. But if you had less than one, if you had more than 0.1 cc's, your median overall survival was months, many months shorter. Now let's switch gears a little bit and look at the TERT mutated MGMT methylated EGFR copy number gain or mutated tumors. And there, it was quite the opposite. We found that the optimal cut point that separated the better survivors from non-survivors was up to 11.7 cc's, right? So basically, in this methylated cohort, patients did really well. If you and obviously there's a little bit of heterogeneity here, in that this cut point, if you kind of reduce it more and more, you probably get better survival. But the optimal survival difference between patient populations was 11.7 cc's. Now that tells me something about this tumor. Like if you're a biologist and you're thinking about this, and you think, well, in one tumor I have 0.1 cc's, meaning that I can leave nothing. In another tumor I have 11.7 cc's, meaning I can leave a little bit more. Then the biology ought to be different. And we've actually done a bit of work that I'm not going to present today, that shows that the immunologic signatures and specific immune cells in these two cohorts of patients are remarkably different. If you have MGMT MGMT unmethylated tumors, as expected, you really also had to resect the entire tumor, and when you did, the survival was a little bit better than average. And then there's this MGMT methylated and p53 altered group that doesn't have the standard mutational profiles of the rest of the tumors. So these cut points, as you can see on the left, were highly variable, and I think this is kind of captured in this slide here, where for one group, right, if you don't get every single bit of tumor out, there's a survival difference of 28.9 months average survival, and these are not small numbers of patients. These are large numbers of patients. So that's getting close to 3 years to what Lucy and I were talking about. I mean, so this group is where you really want to do everything you can, but for the cohort below that cut point, there's only 13.9 months. On the other hand, for EGFR altered and methylated patients, the cut point was 23.8 months, and for when you were above this very high volume of tumor, whereas the cut point below that was 11.3 months, and so these are and then the TERT wild type groups, and obviously MGMT unmethylated groups. These patients do extremely poorly. This is what we think of as glioblastoma, and you do need to resect the entire tumor. So what's emerging is this picture where not just is there genomic heterogeneity, but there are differences in surgical approaches that might follow that genomic heterogeneity. And this is the data just sort of captured overall, where the 3-year survivals were quite different depending on which genomic class you belong to. And if this were a neurosurgical talk, we would sort of talk about some disagreements in the field about the impact of methylation, people finding that that methylation matters in some cohorts, and others finding that methylation doesn't matter. But I think we've kind of teased out the specific cohort where methylation matters. But in the end, I think what is very clear is that when I take my surgical hat off, then now I'm thinking, okay, well, I can get patients to, maybe close to 3 years if I do my absolute best, but that's still not good enough because, really at the end of the day, it's about, the capacity of the tumor to proliferate versus how immunogenic it might be. And in general, most gliomas are non-immunogenic, so it really comes down to the capacity to proliferate. And ultimately, as the tumors proliferate without having adequate therapy, you will get to a point where the tumor will transform and gain, genomic alterations that will make these patients worse. So I'll talk more. So I'll kind of hone in on what I work on, and obviously this is increasingly I work on, a larger swath of GBMs, but really what I started my career on were these IDH mutant tumors, and IDH mutant tumors are really interesting because for me it was kind of like an orphan disease that no one really cared about when very few people cared about when I started. In fact, my chair at Yale thought that there was no reason to operate on these tumors unless the patient seized. He happened to be an epilepsy surgeon, but when you kind of look deeply at the epidemiology of these tumors, you realize that glioblastoma might be an orphanish disease, but low-grade gliomas, because the patients live a little bit longer, in spite of their lower incidence at 1 out of 100,000 persons, the overall person years lived with the tumor is much higher for glioblastoma for diffuse low-grade gliomas with IDH mutations. Now, this is really, I think, is critical because what that means is that the prevalence of the tumor is actually higher than the prevalence of the tumor for glioblastoma. So at any given point, you have more people living with IDH mutant gliomas than you do with glioblastoma because glioblastoma is so bad, so that's because of the survival differences. But so as I was saying earlier to Lucy, I think this is really what drove me to think, well, this is a tumor where we potentially have an impact, the potential to make an impact, because we have a little bit of a runway while we develop therapies and learn about how to approach GBM. So the IDH mutant gliomas are incredibly interesting because they sort of they take everything that I talked about in terms of like the growth factor alterations and cell cycle and oncogenic pathway alterations, and they kind of throw it out the window in a way because this is really an epigenetically mediated tumor. So what happens is that in a normal setting, the isocitrate dehydrogenase would produce alpha-ketoglutarate, which is a cofactor for many of the demethylating enzymes, especially of the TET2 class and other classes of demethylating enzymes. But when you have an IDH mutation, it induces this neometabolite called 2-HG. And what 2-HG does is it binds the alpha-ketoglutarate site on these demethylases with more potency than alpha-ketoglutarate itself does, and so it blocks methylation. And what you get is a non-stochastic sort of methylation pattern. And I say non-stochastic because essentially it recapitulates what a GBM would do, which is to allow for activation of growth factors, block tumor suppressors, and to some extent allow for progression through cell cycle, but all through methylation. So a single enzyme, as a single gene, can do this. And so I found this really, really fascinating because that means that meant that we could really do one-to-one correlative studies with this single mutational event. The other part of this is that, as I talked about before, the average patient with the GBM dies at 59 or so, but all of these patients will die, and the average age of death is 48. So it's not like we can do a lot with surgery. These are surgical patients that I personally and like like my expertise lies in the resection of these tumors, because they're often in highly eloquent areas, and many of the tumors have to be resected with patients awake so we can take out as much as possible. But we can talk about that till we're blue in the face, but all these patients ultimately die. So what do we, how do we sort of address this? And the way I look at it is, it's sort of like the way I started thinking about these tumors is like they are, was there a possibility they're immunologically camouflaged, like this owl in a tree that's camouflaged, for long enough until it can gain the necessary genomic alterations to really become this lion that's kind of, roaring in the jungle that doesn't care if you see it or not, which is what GBM is. It doesn't care if you see it or not. Like you can see it, you can do something about it, and it still will take your life. But these tumors present IDH-mutant tumors present a slightly different pathway. So what I'll talk about in the final part of this talk is talk about, from genomics to immunotherapy, because we, based on what I told you guys about, my initial sort of, scientific influences, this presented an example, the perfect instance to study what the immune microenvironment of these tumors really look like. And so our questions going into this, when I started my lab in 2013, was what immunogenic signatures do IDH mutant gliomas have, and what do they talk about the biology? What does the local immune microenvironment look like? Are there any correlative circulating immune signatures? And this was really, I think that was like a key question, because no one was really looking at correlative circulating immune signatures at the point. And ultimately, how does a genetically weak tumor, if you can think about it as genetically weak, protect itself from natural and synthetic immunity, because these tumors are really highly non-immunogenic. And so we started looking at patients and really resecting large fractions of tumors to do a number of studies, including, starting with basic flow cytometry. And sure enough, what we found is that these tumors are as immune cold as they come. And, what's interesting is that by single cell, genomic analyses, you don't really see this because as clearly, because you really, because deconvolution is not, is quite good, but it doesn't tell you 100% that the cell type that you think is a cell type is actually the cell type. So, using flow cytometry, we could really tell that the immune cells that we care about, which were, natural killer cells and NK cells, were really dramatically lowered in this tumor, which kind of makes sense. Like basically, these tumors don't undergo sufficient immune surveillance. It's like, they're basically like, like there's nothing to see here, just keep going. Whereas if you progress to a high grade GBM, there are lots of perturbations, and it's a different event altogether. But what we also saw that I thought was really fascinating is that we could identify alterations in the peripheral blood that were that really could allow us to predict patients who had IDH mutations using this, cytometric based analyses, where you needed to capture a couple of different subtypes, and they really were all monocytic subtypes. So IDH mutant tumors just happen to have far more monocytes and M2 macrophages, and that was really kind of quite interesting. So now, going back to, and I present this data, and I present it as it is, because it's really the first way we looked at this data. But looking at the TCGA, going back to TCGA, and looking at specifically the immune genes that are present in IDH mutant tumors compared to IDH wild type, what I can tell you is that what you see is an absence of lymphocyte activation, and our initial approach was, well, we're gonna replace these genes, and then we're going to get, but we're going to get an immune response. But as I was talking, when I first gave one of my first talks, while I was at Pitt, one of the, faculty who happened to be chair at the time of immunology said, she said to me, Duca, you realize you can't have immunotherapy without immune cells, right? And you showed all this beautiful data talking about how there's no immune cells in this tumor, but you can't have immunotherapy without immune cells. And so I point out this gene in green, which just happened to be part of the set of immune cells that we looked at when we compared IDH wild type and IDH mutant tumors. But we really didn't know. I mean, we didn't know what it really did. And ultimately, one day I was just reading a paper, and it was this paper by Louis Lanier at Berkeley, where they where they basically talk about the cloning of these NK cell ligands and how they were initially cloned because they were because of exposure to all trans retinoic acid, which we think of as a differentiating drug, right? And when we did this, when in this paper just kind of put it all together because I knew that these NK ligands that are activating ligands that allow a lymphocyte to capture to see a stressed tumor cell were methylated and repressed in IDH mutant tumors, and that this gene called RBP1 was also methylated and repressed, and that this gene, what it does is that it allows retinol or vitamin A to be ultimately turned into retinoic acid. And so RBP1 is a critical chaperone, and it was missing in IDH mutant tumors. Now, what's interesting is if you look across the entire IDH spectrum, across all diseases, there is some form of retinoic acid dysfunction. In fact, if you look at AML, there are no AML tumors that AML events where you have retinoic acid or RARA PML mutations, which is a loss of retinoic acid signaling, and IDH. And if you look at cholangiocarcinoma, it's RBP4, which is another chaperone. So this is really it was non-accidental. And the question was like, okay, well, when we discovered this event was like, well, we thought, well, okay, we'll be able to re-express these ligands that are required for NK cells to kill, even though there are no NK cells in the tumor, right? So we did this. We did a series of experiments, proving this. And in fact, what we found was that we could do this in a genetically engineered murine model. I think it's really important to point out that this was done in an RCAS-TVA model, which is a model where it behaves exactly like the human tumor. It's not very immunogenic. And using this model, I mean, first looking at the human data, we found that the RBP1 locus was in fact methylated, that IDH mutant tumors did not make RBP1. But really stunningly, what we found is that when you look at the amount of retinoic acid by spectroscopy that was produced in IDH wild type versus IDH mutants, I mean, it was a magnitude of 1 to 1,000, thousandfold difference in general. So this meant that we were onto something, but what did it really mean ultimately? And we also showed that retinoic acid dependent transcription was down in IDH mutant tumors, and that we saw that in both in all forms of low-grade gliomas, including astrocytomas and oligodendrogliomas. What was really fascinating was when we then took this into mice. We, along with Eric Holland, I created this IDH mutant genetically engineered model of glioma, which really recapitulated the tumor quite nicely. And then we treated them with retinoic acid. And the first event that happened that was really telling is that the first group of mice that we treated with retinoic acid all died. And when we looked at why they died, I'll show you here, but they died of like cytokine storm, which was unusual, right? And so we then started to go back and lower the dose, and ultimately we found that it was, you know, highly efficacious, and this is in a model of the tumor that, you know, where all the mice will die, as all the humans do. And in this cohort of at least 10 mice, or, you know, ultimately like only one mouse died in the Atra group. But what was more fascinating was that, you know, was what we didn't expect. We did not expect this at all. We expected that the ligands that are controlled by retinoic acid would increase. But what we didn't expect was that you'd have an increase in the total number of immune cells, and that specifically you would get an increase in the overall proportion and overall number and proportion of lymphocytes, both NK cells, CD8 T cells, and importantly CD4 T cells, which, you know, there weren't we don't show that here because there weren't a lot of great CD4 antibodies for this particular mouse. But we ultimately kind of captured that also CD4 T cells are not only important, but they're probably the most important. But this was a whole immunoregulatory event because we also saw a decrease in regulatory T cells, monocytes in general, especially monocytic MDSCs, and then when we looked at peripheral blood, this was recapitulated. So, you know, I like literally, this is one of the experiments I had my lab do so many times because I just didn't believe it. And eventually they're like, you're calling us liars. And I was like, no, I really just want to find out what's going on. But this data, along with some clinical data that I don't show, where we found that patients that were treated with retinoic acid as part of a trial that was meant for glioblastoma, but happened to have IDH mutant tumors, actually, many of them were still alive. And so Steve Bagley and I sort of came up with this concept of a phase 2 trial with retinoic acid plus PD-1 inhibition, and importantly includes a surgical arm and a non-surgical arm. And this is really the ultimate sort of, you know, expression of this of our data, and you know, showing that you could really kind of, you know, like from bench to bedside in neurosurgery is just exceptionally difficult. And remarkably, we have a couple of people at Penn who have done that, which I have a lot of pride in, that this is a place that allows one to do this. But when you look at the human data from the first 7 patients, what we saw was that you were getting these activated M1-like macrophages at later time points after cycle 3 of the retinoic acid, and these were exactly the same type of myeloid cells that we were seeing in the tumor of mice. So now I'll just kind of talk about some very, very, very early data from the trial. We have treated about 16 or 17 patients so far. Of those patients, I think 7 or 8 of them remain on trial. The longest patient who's been on trial has been on trial consecutively for about 20 months now, which, you know, considering how fast our tumor was growing before, that's pretty remarkable. And then there's some patients who perhaps we shouldn't have treated because their disease was so advanced, but it is quite interesting. And what we've found is, again, recapitulating the murine data, which is really interesting, except for the fact that we didn't really look at B cells, because it turns out that B cells in the group of responders is really kind of the ultimate marker of response so far. This just taking 2 random patients who've had up to cycle 9 and comparing them to patients who kind of blew through the Atra PD-1, and showing that, that you have lots of many magnitudes higher B cell fractions. But interestingly, also naive T cells, right? Both CD4 and CD8. And what's really interesting is that we're now starting to build a biomarker bank that we can use to really ultimately understand who might be responding to the trial. And I'll tell you why that matters. The reason it matters is because usually in IDH mutant tumors, in all glioblastoma, one of the biggest events is that we, using basic MRI, we don't really know who is, it's very clear when patients are clearly progressing. But when they are kind of progressing versus not progressing, we have absolutely no idea based on even advanced radiographic methodology. So this is a patient who was treated at Thomas Jefferson initially in 2020, and then had this very robust recurrence, and then ultimately was resected there, and then a month later had another recurrence. And so he was put on the non-surgical trial of non-surgical arm of the ATRA-PD-1 trial. So 4 months post-trial, and I should make one more note that this tumor is located exactly in the expressive speech region. And this patient had absolutely no symptoms, but what they did have was it's really kind of ugly looking enhancement pattern with some changes that we didn't understand, like these sort of cystic changes. So we were obligated to take the patient back to the operating room, and this gave us a chance to take the patient back. And then as an offhand sort of comment, I'd suggest to the pathologist that they look at the number of CD3 cells and also the number of macrophages. And what they found was really quite fascinating. Number one, this patient actually is having to be taken off trial because they have viable cells, and that's one of the weird things about the ethics of trials is that once we take that patient back and we haven't cured them, we basically have to continue them on trial. But what we saw was incredible, in that like I said, IDH mutant tumors are a wasteland for lymphocytes. If you look at any high power field, you might see a single CD3 positive cell. And you can see from the tumor that was resected at Thomas Jefferson, where you have all these primitive looking cells, very, very viable, lots of vascularity. And then in the tumor that we resected, you see a lot of clearing of these tumor cells, and then also normalization of the primitive appearance. But I think what was most fascinating is that in tissue, and we have tissue that we haven't analyzed, but this was just a chance to just kind of look at tissue, off trial, or just a patient who wasn't on the neoadjuvant arm. And so that when they looked at the CD3 proportion, this is by far the most number of CD3 cells I've ever seen in an IDH mutant tumor. And so one can question, what are these CD3 cells? Are they Tregs? Are they, activated CD4 cells? But based on the peripheral data, I think it's quite interesting. At the very least, we know that we have a physiologic event ongoing. So I'll end by just saying that that we're doing some really cool things and are always open to collaboration and looking at novel combinatorial targets. A couple of other trials that we're opening up, and there's a lot of data that I didn't talk about but obviously the point is not to talk about every single piece of data. And with that, I'll thank. There's just there's so many different people to thank, and so many great people at Penn who've been incredible to me in the last 2.5 years since I've gotten here, and allowing me to sort of get my program up and running, and the funding, including the Cancer Moonshot Award that we got, I think 7 or 8 months ago, and other funding from the NIH and multiple foundations, and thank you all for listening.
Thank you so much. I'm sure people have questions and I think we've gone over, so I hope that you will email to ask them. I feel like we're gonna need a yearly update. This is probably the most hopeful thing that I've seen in the last 40 years of um around this work. So thank you so much. And this is so exciting to see even the needle move a little. So thank you, thank you, thank you.
Yep, you're very welcome.